Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Niao, konnichiwa, 안녕하세요, and hello all. And welcome and thank you, SynBYSS people, for joining us, the 109th seminar, and I call it a New York University Day. I embarked on my first 2024 trip to Singapore last Saturday, arriving here last Monday after 32 hours of transportation. It was worth having that long trip because I met a lot of great people discussing great science and research here at National University of Singapore. Thank you all, especially Matthew Chang, for hosting me here. Sadly, it's midnight here, and after this seminar and probably 4 hours of sleep, I should leave this hotel room to catch my flight back to US for another seminar trip to Minnesota, and much colder place. Basically, the temperature change will be from 30 Celsius to below probably minus 10 minimum Celsius, I guess. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Jef Boeke. He does not need any introduction because we all know him. He is a professor and director at Institute for Systems, Genetics, and SynBYOmed, NYU, Langone Health. He is a National Academy member. He's very well known to the research community with his contribution to leading the synthetic yeast genome project with a global collaboration group of people, including, again, Matthew Chang here. Multiple alums or current members will give a talk, his group member, as a rising star later this year. Professor Jef Boeke, thanks so much for your amazing contributions to the scientific community and nurturing young scientists and engineers, and the virtual podium is all yours now. Thank you again.
All right. Well, thank you very much, Tae-Suk, for this invitation to speak to a large community of people interested in synthetic biology. And I prepared a few slides for my talk, so I'm going to share them now. So I'm actually not going to talk today about the yeast project, the yeast 2.0 project, although we had a big batch of papers come out in November about that. But I'm going to talk about some of our mammalian work that we've been doing a lot of for the last 7 or 8 years now. And I'm going to specifically mention a project in which we very simply are writing DNA backwards. And this is one of many projects that are part of our dark matter project. And this is a project that starts over here on a computer with an idea to study a certain mammalian gene, typically a human gene, and but study it in its very complete and large form, what we call an assemblon, where we can assemble a very large DNA on the order of 100 to 1,000 kilobases. And we put those pieces together in our workhorse yeast Saccharomyces cerevisiae. And we call these DNAs assemblons, but we also call them payloads because they're going to be payloads that are eventually delivered to a highly engineered mouse embryonic stem cell into which we've installed a custom landing pad that will direct exactly where this payload is going to go in the mouse genome. And then because these are now engineered embryonic stem cells, we could differentiate those in vitro, or we could actually turn them into an animal using a method called tetraploid complementation. And then, of course, we can study those cells or animals in a multitude of different ways using genomic methods or behavior methods or anything you can think of. And then we can do another round. So it's sort of a design, build, test cycle, but at the level of potentially a whole organism. So the really synthetic story that I'm going to very briefly cover today involves the human HPRT1 locus, 101,000 base pairs of human DNA, which we have delivered to embryonic stem cells already several years ago. But Brendan Camalato here, who I think will address this group later this year, built not only HPRT1, but also literally synthesized it backwards, so reversed but not complemented. This is an error often made by beginning synthetic biologists and beginning molecular biologists, but we actually did this intentionally. And the idea is when you write something backwards, like this phrase, it's really hard to recognize. And it's essentially random, almost randomized, but not completely randomized. It would still have the same number of G's, A's, T's, and C's as the original sequence. Homopolymer runs would be conserved, but the real logic or sense that's embedded in the HPRT1 gene would be completely destroyed. And then we have the ability now to easily deliver that DNA into either cerevisiae or mouse embryonic stem cells. And this is exactly what Brendan Camalato did. So I'm going to show you now what happens when the forward HPRT1 gene is inserted into one of the yeast chromosomes. And of course, human genes and mouse genes too are huge. They have huge introns, many exons. So this is the HPRT1 gene occupying about 35 or 40,000 of the 101,000 base pairs. And these tiny lines and boxes here are the native yeast genes, which mostly have no introns, and they're just so small that on this diagram, they're all overlapping. You can't even see them all, but they're really tiny. So the genome structure between yeast and mouse or human, dramatically different. But what I want you to pay attention to is these tracks here. These tracks show a TAC-seq, which is going to look for open chromatin, active chromatin. And here's a histone mark of active chromatin. And here's actually RNA-seq track and a cage-seq track, which basically maps 5' ends of RNAs. And what you can see is that in this shaded region, which is the human region, there's a multitude of cage-seq peaks and more evidence of open chromatin and RNA in the human DNA than there is in the flanking yeast DNA, which was a somewhat unexpected result. Now, when we did the same thing with the backwards HPRT, we actually got a very similar result. A lot of ATAC-seq and H3K4 trimethyl, RNAs, and cage-seq peaks way more than what we see in the flanking yeast DNA. So what happens when we take this backwards HPRT and put it into the mouse? Well, in mouse embryonic stem cells, we see exactly the opposite result. We see almost no ATAC-seq peaks, no H3K4 trimethyl, very little evidence of RNA polymerase, and no RNA. Furthermore, we see evidence of a silencing type of structure with H3K27 trimethyl in the more GC-rich portions of this HPRT1 reverse gene. So how do we interpret all this? Well, it seems that essentially random sequences that yeast has never seen before are very actively transcribed. And Brendan even showed that they can be translated if connected to the appropriate yeast reporter. The transcripts are short, but very, very abundant. And it suggests that the default mode for inserting random sequences in yeast is that they're very highly expressed. So we actually think that most of the yeast genome is actually under selection for the lack of spurious transcription so that the proper genes can be expressed. And needless to say, random sequences would seem then to be fertile ground for the production of brand new genes. And we think this may be how new genes are actually born. And most surprisingly of all, in the mouse embryonic stem cell, we see that most of the sequences seem to be totally shut down for expression. And even if we remove all of the CpG sequences, which are methylated in the mouse, we still see that these are completely shut down for expression. So it suggests that the default mode for gene expression is fundamentally different between yeast cells and animal cells. And we asked Dolly 2 what it thought about this. And the prompt that we gave it was an out-of-control yeast meets a buttoned-down mouse at a DNA party. So where are we going with all this? Well, here are a couple of things that we are thinking about and working on. I've been a natural history buff for all of my life, and this is a picture of the great auk, a species which went extinct in the late 1800s. It's an iconic species, here pictured in a print by John James Audubon. And of course, it is theoretically possible to resurrect extinct species DNA sequences and potentially to insert those into a closely related species and imagine bringing back an extinct species. Should we do that? That's an important question that requires discussion. A second thing that we're thinking about is whether we can do neuroengineering. And in particular, we're interested in this bird and a unique trait that it has, which I hope you can hear.
Never shake a baby bird. That would surely be absurd.
Now, why did I show you that picture? Well, this is Disco the parakeet, and Disco is remarkably able to mimic the sounds that he's been trained to remember and repeat. And so this bird, with its tiny brain, has prodigious vocal learning capabilities, similar to us and a small subset of the organisms on planet Earth. Eric Jarvis has looked at gene conservation and gene expression patterns in the parts of the brain that control vocal learning. and has identified 5 human genes that are implicated in vocal learning. So we are currently working with Eric and constructing big DNA, the relevant big DNAs, for delivery to mouse embryonic stem cells. And Eric has the tools to evaluate whether the mice that get those genes have improved vocal learning capabilities. And with that, I will stop and thank my amazing team and my collaborators around the world, as well as at NYU, for their amazing participation and leadership in all of these projects. And with that, I will stop.
Thank you so much. It's amazing, and I'm fully awake now, and because of your amazing talk. And it is also coincidence. I went to Chinatown yesterday, and then I saw the amazing bird parrot, and then probably she or he speak Chinese much better than myself, and I'm amazed by that. Thank you so much for your wonderful talk.
My pleasure.
Okay, thank you. Now, the main speaker of today with a longer introduction, Dr. Tetsuhiro Harimoto received his PhD in biomedical engineering in Dr. Tal Danino's lab at Columbia University, basically the same city with the Jef Boeke currently located. And he got the degree in 2022. His graduate work centered on engineering living microbes as drug delivery systems. In particular, he developed synthetic biology and bioengineering technologies to engineer bacteria capable of sensing and delivering therapeutics to solid tumors. Currently, he is a postdoctoral fellow at the Wyss Institute for Biologically Inspired Engineering at Harvard University. Working with Dr. Dave Mooney, he is designing next-generation living drug delivery systems. He has been named as MIT Technology Review's Innovators under 35 and received multiple awards, including the NCI Pre-Doctoral to Postdoctoral Fellow Transition Award, so-called F99K00, and he is truly a rising star with great potential to become a future leader. If you are a faculty search committee member, you do not want to miss him. Tetsuhiro, thank you so much for your time today, and please take it away. It's all yours now. Thank you.
Thanks, Tae-sok, for the kind introduction, and thanks, Jeff, for giving an inspiring talk earlier. I've been following the SynBYSS community for a while, and it's really great to see this community coming together, and I'm really excited to have a opportunity to talk here today. So let me share my screen. All right. Great. So today I thought I would talk a little bit about what I've been working on for the past years, specifically on engineering living bacteria as a way to deliver drugs to tumors, and how we could utilize synthetic biology tool sets to turn these bacteria into the next generation of living medicine. So the biomedical community in the past couple of decades had really created many new exciting drugs that has potential to solve and treat many diseases. But in the current medicine, one of the grand challenge is to deliver these drugs to specific sites in the body at the precise timing and dosage. And it's been very difficult to do so. So I thought I would start by sort of listing out some of the problems that we face today in terms of drug delivery. So let's say we have a tumor that we want to deliver drugs to, and we would be administering drugs, whether it's chemotherapy, immunotherapy, or whatsoever. Upon initial injections, many of these drugs actually get absorbed by the surrounding tissue and gets lost, which leaves us quite a, which leads to significant degradation of these active compounds. And even if some of them gets to the tumor sites, many of them might have a difficulty penetrating through tumor tissues because of their dense matrix and pressurized environment. And above all, many of these drugs have low stabilities in the complex physiological environment, and therefore giving us a very narrow time windows in which to act upon. So what's the outcome for these type of limitations? Well, we're experiencing poor efficacy, and attempts to increase the efficacy oftentimes necessitates high dosing or repeated injections, which ultimately leads to toxicity. So I'm looking at this problem from a very different perspective, where here what we're looking at are the bacteria that naturally colonize wide range of physiological niches in our body. So these includes bacteria that colonize the gut or on the skin. And more recently, we've been discovering a different type of bacteria that can colonize solid tumor specifically. So now these microbiomes are naturally introduced from surrounding environments. As bioengineers, we wonder whether we could now intentionally administer these type of bacteria so that they would specifically colonize tumor in this case, and perhaps engineer them so that they could produce drugs from within these tumor tissues. And by utilizing synthetic biology tool sets, we could have them precisely deliver drugs at the right timing and dosage that's required to treat these complex diseases. So how does these bacteria colonize tumor in the first place? Well, the mechanism is still under the investigation. There are critical steps that we think is important. First, these bacteria gets deposited to the tumor tissue through leaky vasculature, where they are protected from immune clearance due to the immunoprivileged environment that many solid tumor provides. And after some time, these bacteria will proliferate and colonize these necrotic core of these tumor, thanks to the physical space and nutrients that's produced from these dying cells. Now, what you're looking at here is a picture of a mouse that's colonized by the bioluminescent E. coli upon intravenous injection of these bacteria into a mouse that has subcutaneous hind flank tumors. So I've taken this picture after about a day or so, and we could already see a very high specific signal that's coming from these tumor environment. Now, we and others have shown that there are different types of bacteria that can do this, including E. coli and Salmonella that are more genetically tractable. So the idea of utilizing bacteria for cancer therapy actually isn't that new. And if you go back in history, one of the first reported case was reported about more than 100 years ago by a doctor at the time in New York who've injected live cocktails of bacteria into sarcoma patients. So they've actually shown pretty remarkable result where they show up to 60% of response rate. But one of the problems here is that they've used unmodified pathogenic bacteria even before the time in which the antibiotics are widely used in the clinics. So unfortunately, this approach wasn't deemed sort of viable at the time. Now, moving for about almost a century, there are a small number of clinicians who started clinical trials to try to utilize these bacteria for cancer therapy. Now, utilizing attenuated form of bacteria. So what do we get from these clinical trials? Well, we've shown that these attenuated bacteria could indeed colonize human tumors, which are pretty exciting. However, there was no efficacy that was observed in these clinical trials, likely due to severe attenuation of these bacterial strains. So currently, the field has been focusing on the engineering of these bacteria. And the idea here is that if you could have these bacteria deliver payloads, then we could improve the efficacy. However, one of the problem here is that there has been very limited methods to control these behavior of living bacteria in a complex in vivo environments. And therefore, it's been very difficult to bring this technology towards clinical translations. So right around when I joined graduate school, synthetic biology as a field was really maturing, including many of the work that's been done in this community. And I don't think I need to, explain many of the basics here. But basically, the idea here is that we could construct synthetic genetic circuits that we could transform into living cells, whether it's bacteria or mammalian cells, to have them produce new type of functionalities. So in this case, we could, transform bacteria with the gene encoding for antimicrobial, anti-cancer proteins, and have these bacteria kill cancer cells. And on top of that, we could couple that with biosensing capability so that these bacteria can now sense and respond to different environments. And by applying these type of therapeutic bacteria in vivo, I believe that we could solve many of the challenges that I've described earlier. So for the rest of the time, I wanted to introduce 3 different stories in which we're trying to push the boundary of these therapeutic bacteria for clinical translations. I'm going to talk about how we're thinking about delivering these bacteria more efficiently. I'm also going to talk about some of the in vitro platform that we built so that we could screen for these therapeutic bacteria more quickly. And I will end up by talking about some of the strategy that we're thinking about in controlling bacterial behavior and their specificity to tumors using synthetic gene circuits. So let me start by talking about the delivery of these therapeutic bacteria, specifically utilizing some encapsulation strategies. And this is the work that was done with a postdoc at the time in the Danilo lab, Jason. So when we think about translating these type of bacterial therapeutics, toxicity is really one of the major challenge here. So to illustrate, I basically plotted a very simple schematic where the y-axis is toxicity and x-axis time. So initially, upon injection of these bacteria, a patient would experience a severe toxicity due to the high immunogenicity that the bacteria inherently has. So this could lead to cytokine storms and other toxicity that would be deemed dangerous. So more recently, researchers have been thinking about this problem, and one of the emerging technology is to encapsulate these bacteria in some kind of inert biomaterials, so that these bacteria can now evade immune systems. Well, one of the downside of these type of approach is that if you could over-evade these immune systems, then the bacteria could linger around in our body for a prolonged time and potentially could help them, grow uncontrollably, which would also lead to toxicity. So our idea is to simply think about a way in which we could transiently encapsulate these bacteria, and we could turn them off when we don't want them to linger around in the body anymore. And we turn to synthetic biology toolsets so that we could dynamically regulate these bacterial surface properties. So the structure that we decided to focus on is called capsular polysaccharides. And here, what I'm showing is a cross-section of an E. coli probiotic strain called Nissle. That's going to be a focus of this study. So what you're looking at are these fuzzy structure composed of sugar molecules that physically coats the outer surface of these bacteria. And these capsular polysaccharides are known to be important in protecting these bacteria from environmental threats, including antimicrobials and immune clearance. So we wanted to find a way to engineer the expression of these capsular polysaccharides so that it's going to be under our control. So to do so, we simply created libraries of knockdown strains, each targeting the biosynthesis pathway of capsular polysaccharides. We found one of the gene KFIC to be critical in producing these capsules. Now we simply put back the KFIC gene on the plasmid, but under a repressor molecule called LAC-I. So only when in the presence of inducible molecule IPTG these bacteria would be able to produce the capsule polysaccharide components called heparazine, which consists of the capsule polysaccharide components. So how does this look under the microscope? On the left, what I'm showing is a cross-section of bacteria in the absence of IPTG. And on the right are the bacteria that's fed with IPTG. And what we saw was a significant thickening of this bacterial outer surface structures, indicating that we can now control the expression of these capsular polysaccharides. So we decided to call this system as inducible cap, or in short, ICAP. And by expressing these ICAP, we could drive these bacteria towards immune evasion, or we could simply turn them off to facilitate the clearance of bacteria in vivo. So we were pretty excited about this system, and we wanted to first characterize to see if we could modulate the bacterial interaction with different components of immune systems. So we first look at the interaction with humoral immunity. So in this experiment, what we did is to expose these ICAP strains with and without IPTG in human blood, which contains many of the humoral immunity components such as complement. So here's one of the results, where on the y-axis is bacterial viability in the blood over time on x-axis. So our positive control, which is the ECN wild type, which always has a capsule, would linger around in the body, in the blood for quite some time. Whereas the negative control, del KFIC, that doesn't have any capsule, would rapidly die inside of these blood. Now, utilizing ICAP system, we could basically modulate the time in which these bacteria would survive in the body. And we could utilize these type of phenomena to really take advantage in controlling pharmacokinetics of bacteria in circulations in vivo. We also look at their interaction with cellular immunity, in this case, macrophage phagocytosis. So what we did is to expose these bacteria to phagocytic macrophage and see how much of a bacteria gets recognized and gets phagocytosed. So here what I'm showing is a number of bacteria that gets phagocytosed in the macrophage. And we show that the activation of ICAP strain could really bring down the phagocytosis by about tenfold. We also looked at the immunogenicity of these bacteria by exposing them to monocytes and looking at their cytokine productions. So here I'm showing one of the representative cytokines, TNF-alpha. And again, we show that the activation of ICAP could reduce their immunogenicity compared to the controls. So now we've shown that we could modulate their interaction with the immune system. We wanted to go back to the initial sort of motivation and really ask whether this ICAP system could be utilized to improve the toxicity, the safety of these bacterial therapy. So here we're moving into the in vivo system, where we're intravenously injecting these bacteria to a mouse, and we wanted to look at their toxicity. So a very brief recap, we have two controls. One is a del KFIC that doesn't have any capsule, that's immunogenic, and the wild type E. coli that's not immunogenic, but would linger around in our body for quite a while. And the ICAP strain that we initially induce with IPTG before injection, but we don't provide IPTG after we inject them, and then we expect them to lose the capsule after some time. Now, when we look at the toxicity represented by the percent weight change in the mouse, we saw that the ICAP strain performed better as compared to the control. But what we really wanted to ask is whether we could really establish a relationship between the bacterial dosage and the toxicity. So what we did is to repeat this experiment, but in a dose escalation manner. So that result is shown on the right, where on the y-axis is showing the toxicity level that the mouse had experienced, and on the x-axis is the increasing dose. So for the two controls, as we increase the dose of the bacteria, the mouse would experience more toxicity as expected. But when we studied ICAP, the dose toxicity curve seemed to have shifted towards the right. And from these curves, we could calculate the maximum tolerable dose. And we determined that the NTD for ICAP is about tenfold higher than the controls. So in other words, we can now inject 10 times more bacteria without sacrificing their safety. So the question that we wanted to ask is whether we can now improve the therapeutic efficacy. So what we did here is to inject these bacteria into the mouse, but now these mouse has tumors. We further engineer these bacteria to now produce therapeutic payloads, in this case, theta toxin, which is pore-forming toxin that's known to kill tumor cells. And then we injected our three types of bacteria at their maximum tolerable dose. So here, ICAP strain 10 times more compared to some of the controls. So what I'm showing on the right is the tumor growth over time. And what we were able to show is that the ICAP strain at the maximum tolerable dose was able to slow down the growth of the aggressive tumor model, in this case colorectal cancer, by far as compared to some of the control strain. And one of the things that I wanted to emphasize is that even though these guys are injected with different amount of bacteria, their safety profile basically remained the same. And we show the therapeutic efficacy, not only just the colorectal cancer, but also in different orthotopic models, including breast cancers. Okay, so I hope I was able to show some of the examples in which we're thinking about delivering these bacteria to patients. But once these bacteria gets to the tumor, we want to make sure that these bacteria kill tumor cells very effectively. So that leads to our next story, where we've established a co-culture system to really try to maximize the efficacy. So when we think about designing these bacterial therapies, there are multiple parameters that we want to engineer. Let's say, what are the tumor cells that we're going after? What are the bacterial strains or species that we want to use? And on top of that, what are the circuits and genetic payloads that we want to add to these bacteria? So this vast combinatorial space is very difficult to screen using existing animal models. So to circumvent this problem, we developed a new in vitro system in which we can mimic the bacterial colonization within the 3D tumor model. And by doing so, we can now screen different therapeutics. But not only that, we could also look at other different characteristics, including genetic circuit dynamics and multi-population interactions. So we chose to utilize 3D tumor spheroids as a model system because of a couple of reasons. One is that these tumor spheroids are known to create a necrotic core, which allow these bacteria to grow within. These necrotic cores are also known to have hypoxic and acidic environment that are important for these bacterial colonization. And most importantly, we could produce these tumor spheroids in a homogeneous manner in a massively parallel multi-well plates so that we could utilize this for high throughput screening. So to establish this system, what I did is to create these tumor spheroids. And as a naive, first-year grad student, I added bacteria on top of it, hoping that they would just colonize. But what I quickly realized, as shown in this movie, is that what I'm showing here is the tumor spheroids in a circle and the bacteria expressing GFP here. And what I showed was that these bacteria would quickly grow into these culture media, making it very difficult to establish co-culture. So in hindsight, this is not very surprising because in vivo, these bacteria are sort of contained in the tumor because when they try to come out, there are immune systems that clear them away. So I wanted to mimic that system. And our strategy is to screen different type of antimicrobials that could kill bacteria outside of the tumor spheres. So after a bunch of screening, we found that there's a particular antibiotic called gentamicin that would kill bacteria effectively outside of the tumor spheroids, but it'll have very poor diffusibility, and therefore they could spare the one that's inside. So we basically incorporated gentamicin into our protocol, and now we could get these bacteria to selectively invade into the tumor spheroid over time. Okay, so with this system, we can now get these growing population of bacteria specifically contained inside of these necrotic tumors, necrotic sites in the tumor spheroids, and we could stably co-culture these two populations over days to weeks. So now that we have this platform technology, what can we do? Well, we can do a lot of things. For example, screening different parameters, including genetic circuits, therapeutics, and bacterial species, and pick the best one and validate them in vivo. So I'm going to quickly show one of the things that we did, which is on the therapeutic screening. So here what I'm showing is a bacteria that's engineered to produce different therapeutics under very simple inducible systems. So what I'm showing on the bottom is the tumor spheroid growth on the y-axis, tested against 10 different types of therapeutic payloads. Now we show that there are several therapeutic payloads that works better than the others. But what's cool about this is that many of these therapeutics has been tested individually in previous papers, but they've never been compared one against another. So this really shows some of the things that we could start to do by comparing on the same sort of platform how these therapeutics would work. But more excitingly, we could start to ask questions about how does these payloads crosstalk with genetics gene circuits. So for example, on the right, what I'm showing is another set of gene circuits called synchronized lysis. So very briefly, these bacteria could grow to a certain population and they could lyse and pop themselves open. And the reason to do this is so that we could facilitate better delivery of these therapeutics from the cytosolic space of bacteria to extracellular space. But when we think about this lysis circuit also sort of reduce the fitness of these bacteria because they would be popping open and effectively killing themselves. So we weren't sure how these synchronized lysis system would affect the therapeutic efficacy. So what we did is to simply clone the 10 same therapeutics under the synchronized lysis circuit and look at their therapeutic efficacy. What we found was rather surprising that we show that these gene circuit to payloads are basically context dependent. So there are some therapeutics that worked better when we couple with synchronized lysis, whereas there's other ones that didn't work better. So while we're still sort of looking into the reason why this had happened, I think this kind of highlights some of the unique things that we could do with these type of co-culture systems. Okay. So now that we could have these bacteria produce potentially very cytotoxic molecules, we want to make sure that these bacteria wouldn't produce them and activate other places other than tumors. So I'm going to wrap up the talk by quickly talking about our efforts to really improve the specificity of these bacteria to tumor utilizing biosensors. And this is the work that was done in collaboration with graduate student Tiffany. Okay, so as I described earlier, when we inject bacteria into our systems, they would colonize the tumor. But because it's systemic injections, they would also land into other organs, such as liver or intestine in this case. So here on the right, what I'm showing is one of the bacterial CFU and their viability in different organs after a day of injections. So what I saw was that these bacteria could colonize tumors at very high level, but in other healthy organs such as spleen and liver, they could still hang around for a couple of days before they get cleared out by the immune systems. So one way to address this problem is to have these bacteria specifically target tumor environment. So in the past, people have utilized anaerobic bacteria that would only grow in hypoxic environment, which is one of the signature for tumors. However, one of the problem here is that there could be some other organs, such as intestine in this case, that's also hypoxic. So we thought, what if we could have these bacteria sense multiple environments, let's say both hypoxia and high lactate, and utilize gene circuits to have them compute what they see and improve the specificity. And by utilizing the same architecture, we could basically modularly combine different type of biosensors to specify where they should grow in our body. So to this end, we developed genetic biosensors. And here on the left, what I'm showing is hypoxic sensor. And very briefly, this promoter PPFT is regulated by the dimerization of these protein called FNR that only activates the promoter in the absence of oxygen. So we characterize this by looking at the GFP expression under different conditions, and the data is summarized on the right. So let me walk you through how this data is read. So these three big boxes basically represents different percentage of oxygen. So on the left is 0% oxygen, on the right is 20%, which is pneumoxic conditions. Now the color indicates the activation of these sensors, and therefore, and therefore what we see here on the left is that these bacteria, these sensors are actually turned on specifically under hypoxic conditions. Now these small boxes represent different amount of pH and different amount of lactate. And what we wanted to show here is that these hypoxic sensors are largely independent of these pH or lactate conditions. So similarly, we built lactate sensor, which showed lactate specific activation, and the pH sensor, which could get activated under low pH. So all in all, what we showed here is that we built three different sensors that could get activated specifically at their specified conditions, and they're largely orthogonal to each other, suggesting their possibility to multiplex. So now we want to control bacterial growth using these biosensors. So we simply put this in front of an essential gene called ASD. So only when these bacteria was able to express this ASD gene, they'll be able to grow. So for this audience, I think we all know that any genetic circuits require multiple optimization. So we basically built a library of different optimized systems with different RBSs, regulators, and origin replications to really hit the right amount of these gene expression so that it's not either leaky or too tight. So let me demonstrate one of the very simple example where what we're showing on the y-axis is the preferential growth of bacteria in hypoxic conditions. So for wild type, they could grow both conditions, so there's not much of a preferential growth. If you put the hypoxic construct on the multi-copy plasmids, then these bacteria would still be able to grow in both hypoxia and pneumoxia because the expression level is probably too leaky. So to reduce the leakiness, we basically reduce the number of copy number into 1 by genomically integrating this cassette. And now we could have these bacteria only grow in hypoxic conditions. Now, as I promised, we've also made a multiplex sensor by combining the PPFT and hypoxia sensor that I've described earlier with lactate sensor. So in this case, the basic architecture remains the same, but instead of expressing the ASD gene, we express another orthogonal essential gene called GMS. So only when they sense both hypoxia and lactate, these bacteria should be able to grow. And indeed, that would be the case. So this data shows the bacterial viability on the y-axis and x-axis different amount of different conditions spiked in with lactate, hypoxia, or both of the conditions. And what we show was that these engineered bacteria was only able to grow when we provide both of the conditions, but not one or the other. So now we take this strain and then inject it into animal model. And after 2 days or so, we take out organs and see how much of bacteria could be found. Now, looking at the tumor, there are a bunch of bacteria, engineered bacteria shown on the right that grew in the tumor almost to the same extent to the wild type, whereas the negative control that doesn't have any of these genes cannot grow at all. But more excitingly, when we look at the healthy organs that doesn't have any of these tumor environment, these engineered bacteria had shown significantly less amount of viability as compared to the wild type, to about 100-fold. So this result really shows that we could utilize these type of engineered bacterial circuits to really improve the specificity of bacteria to tumors in vivo. Okay, So with that, I hope I was able to show some of the ways we're thinking about or we're trying to push the boundary of these bacterial therapies. I talked about some of the delivery methods so that these bacteria could go to the tumor more efficiently. Now, utilizing these co-culture model, we could have these bacteria express very potent therapeutic payloads, and we could incorporate these type of sensors and genetic circuits so that they can only do that in the tumor environment. And through these process, we're building a sort of multi-scale framework in which we could really engineer these bacterial therapies, spanning from genetic circuits all the way to validation in advanced animal models to really bring this technology to clinical translations. Okay, so with that, I'd like to thank my lab, the Nino Lab at Columbia, where I did most of my work during my PhD. I also wanted to thank the Muni Lab at Harvard, where I'm currently a postdoc. I hope I have an opportunity to talk about more of my recent work in the future, my collaborator, both in Columbia and Harvard, as well as my findings. And thank you so much for listening.
Amazing talk. Wow, that's very exciting. Especially because I'm a little bit biased because the last part of the talk is probably my favorite, I guess. Jeff need to leave, but he made comment and question. So he said,
Okay.
very nice talk. The natural tumor microbiome sound like an ideal vehicle. Can you tell us more about which species are in there and whether E. coli is one of them? And I think that's first question. And second question is how will you go about? Okay, let's start with the first question. Can you? And then okay, let me read the other one. How will you go
Sounds good.
about finding the ideal species for delivery of therapies, and will you choose a single species or will you try to engineer a consortium of bacteria species? Interesting.
Yeah, thanks for the question. So let me try to answer the first question. So in terms of the type of bacteria that's found in the natural microbiome, as I recall, it really depends on the different type of tumors that we're looking at. So the one that that's very well known is colorectal tumors. It's not too surprising because it's exposed to different type of bacteria all the time. Let's say Fusobacterium are very much well known to cause and promote these tumor growths, and they exist specifically in these tumor environments. Now, looking at other microbiome in other type of cancer, there could be a lot of different ones, including pancreatic cancer or breast cancer, even lung cancer. But I think one of the things that I wanted to bring up is that the absolute abundance of these bacteria are much less as compared to our engineered strains. So, for example, in our system in vivo, when we inject these bacteria to the tumor, they could grow up to like billions of CFUs per gram of tumors, which is way higher than the natural microbiome that we could find. So the relationship between the natural microbiome and the engineered strains could be quite different by nature, but still, utilizing these natural microbiome as a delivery vehicles to tumor would be a very exciting sort of avenue to pursue. So just to clarify that, whether we could find E. coli in these microbiomes, sometimes we could, but I think it really depends on the context as I described before. So another thing, so the second question was about whether so the second question was sorry, what was the second question again?
Second question is in the chat, but I will read that again. So second question is how will you go about finding ideal species for delivery of therapies, and will you choose a single species or will you try to engineer a consortium of the bacterial species?
Right. Yeah, so that's a really interesting question.
Yeah.
So far, what we've done are single species, and that comes from the fact that it's much more simple to deliver bacteria with single vehicles and single cassettes. However, the future of these like therapeutic bacterial delivery really entails some of the multi-species communities. Even in our efforts, let's say we have 3 different therapeutics that we want to express. We could and we have tried, mixing 3 different population of bacteria and inject them together. And that has multiple advantages, including some of the metabolic burden. You could lessen those. It's also much easier to independently develop strains and just simply mix. So from logistical standards, it's also it also could make sense. Now there are a couple of efforts to really push the frontiers of these multi-species bacterial therapy. For example, some of the group at UCSD have shown that you can have 3 different species of or like a community of bacteria, each killing one or the other. So the idea there is that we could inject, let's say, strain A first, and then you can inject strain B next, and then the strain B is going to replace strain A. We could inject strain C, which is going to replace strain B. And the idea behind it is that even if some of the bacteria, breaks the circuit, lose plasmid or whatsoever, we could still replace these population and sort of have a reset. So there are multiple, exciting ideas that's around the sort of multi-species communities. But yeah, I think those are yet to come.
Okay, exciting. So let me quickly ask one quick question, and then let's wrap up. So now, as you know, I mean, you are the expert, but I'm not in terms of cancer, area. But to me, a lot of years, we invest a lot of money to, conquer, cancer, but still we need to see more. And then, but at the same time, it's kind of very promising because now we have the multiple options, not only surgery or some other, some invasive, the treatment, but now we are talking about, bacterial, killing, tumor cell. I mean, this idea is, for a while, as you mentioned, now, because we have the more options for the doctors, the doctor should kind of choose between among those kind of different options. So what would be, your kind of insight, among those options, how, doctors should choose among these different options? I mean, you are not the doctor, so you may not know the answer, but I just wanted to hear from you anyway.
Yeah, that's a great question. So maybe I'll frame it in 2 different ways. One is sort of like how different bacteria is compared to other, let's say, living therapeutics. And second is how do we think about bacterial therapy fitting in terms of like a lot of different therapeutic vehicles. So the first is that bacteria is quite different from other cellular therapy. I think one of the striking difference is that while other cellular therapies, including like CAR Ts, oncolytic virus, even like antibodies, they all target surface markers. So you would target, HER2 or whatsoever. Whereas for these bacteria, they rely on the tumor microenvironment. So not necessarily a specific marker that's there out on the surface of cancer cells, but more about, the necrotic core and the hypoxic environment that's more universal to different type of tumors. So in other words, it's quite modular. And in our lab, we've shown these bacterial therapy can really be applied to a wide range of tumors, including colorectal, breast, lung, liver, as long as there are these tumor microenvironments, which are quite a fundamental sort of phenomenon for many of the solid tumors. So I do think that's quite a unique property that bacteria can provide on top of many of the other unique things like small sizes, the intrinsic immunogenicity and so on. But the way I'm seeing how these bacterial therapy could fit into the wider sort of like more over like different options of these therapeutics, is that it could really be, something that could be combined with other therapies. It doesn't necessarily have to be that, like, oh, because you're going for bacterial therapy, you can't really use the other ones. And in fact, the sort of future direction would really involve, how do we interact with other therapeutics? For example, people have shown that these bacterial therapy could really synergize with chemotherapy. Let's say these bacteria, just topologically speaking, they're colonized in the necrotic core where they're producing the therapeutics, but the therapeutics might not reach the outside of the cancer. Whereas the chemotherapy is actually the other way around, where they're killing the tumor cells from outside, but it's really difficult to get into the inner core. So by combining those two, we could get synergistic effect. We could also start to think about how they could sort of interact with different population of cellular therapy. So like one of the recent study from Tao's lab shows that these tumor specific bacteria can now produce antigen that could be recognized by CAR T cells. And now these we are starting to talk about these multi-population of bacteria and CAR T cells, sort of like inter-kingdom population, interactions that's working together to kill these cancer cells. So there are quite exciting possibilities that we could combine for these type of therapies, and we're starting to just scratch the surface. But I think there could be a lot more exciting stuff coming.
That's wonderful, John. I'm so excited because that somebody should conquer the cancer all together. We all together eliminate entire cancer in one day. So thank you so much. So let me double check whether we have some question from audience. Okay, I didn't see anything. Okay, they want me to sleep, I guess. So let's close. So thank you all for joining and staying today, and we'll meet again on January 25, Thursday, the same time, the same Zoom link. We'll have Dr. Lynn Rothschild at NASA and Dr. Cameron Glascock at University of Washington in Seattle. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. Thank you, and I will stop recording. Give me 1 second.